Hello everyone. Today we are going to explore one of the most fundamental biochemical pathways in all living organisms that is called glycolysis. Whether you are a bacteria, a plant or a human, your cell rely on this process to extract energy from glucose. Now, imagine you just ate a meal rich in carbohydrate. What happened next? Your body breaks down those carbohydrates into glucose, and glycolysis is the very first step in converting the glucose into usable energy. It's universal and ancient metabolic pathway, meaning it has been around for billions of years and occurs in virtually every cell, from the simplest cell, that is bacteria, to the most complex human tissue. So, why is glycolysis so important? Because it provides the cell with quick energy in form of ATP. 2. It does not require oxygen, meaning it can function in both aerobic and anaerobic conditions. 3. It generates key metabolic intermediate used in other pathways like the Krebs cycle and fermentation. So, today we'll break down glycolysis step by step, exploring how a single molecule of glucose is transformed into pyruvate while also producing ATP, adenosine triphosphate, and NADH reduced form of NAD. By the end of lesson, you will understand not just how glycolysis works, but why it's essential for life itself. Okay, we can start with glycolysis. Glycolysis is a 10-step biochemical pathway that you see in this image that occur or take place in the cytoplasm of the cell in all cells, from simplest to more complex, from a bacteria to human cell, involve breaking down one glucose molecule. So this process initiates with one glucose into two molecular of pyruvate, producing ATP, which cells use as energy because ATP is a common form of energy in the cell. So, this process does not require oxygen, meaning it is an anaerobic process and can occur in both aerobic and anaerobic conditions. Yes, it's true that glycolysis is an anaerobic process, but can occur also in aerobic condition. So now let's go through glycolysis step by step. It is divided into two phases, two principal phases. Okay, phase 1, energy investment, and the second, phosphorylation. So phosphorylation key regulatory step. We can see in this image one molecule of glucose that the formula of glucose is C6H12O6. So glucose is an organic molecule. So it means it contains carbon-hydrogen bond that can store a lot of energy in their bonds. So in the first step, we have glucose phosphorylation. There is an enzyme that is called hexokinase. Hexokinase is an enzyme that phosphorylates the substrate that here is glucose to glucose 6 phosphate by using and consuming an ATP molecule. So, the enzyme hexokinase attach a phosphate group from ATP to glucose, converting glucose into glucose 6 phosphate. That we can say G6P. And in this image and in this PDF, you can see the structural formula as ring of glucose. That phosphate group is added to 6 carbon of glucose and produce glucose 6 phosphate. This prevents glucose from leaving the cell. So, glucose enter the cell and phosphorylated for preventing glucose from leaving the cell. The second stage is isomerization. Isomerization that occurs by another enzyme that is called isomerase, but complete name phosphoglucose isomerase. So this means the enzyme that is called phosphoglucose isomerase rearrange the molecule converting G6P into fructose 6 phosphate because glucose and fructose they have same molecular formula C6H12O6 fructose glucose because they are isomers and they are hexose. Second phosphorylation key regulatory step, the enzyme that is called phosphofructokinase uses another ATP to add a second phosphate to fructose 6 phosphate and producing fructose 1,6 bisphosphate. This means 2 phosphate group in this molecule is added to carbon of fructose molecule. This step is very important and is the main control point of glycolysis. So if the cell does not need more energy, phosphofructokinase slows down glycolysis. After that, we have 4 steps. In the 4th step, the enzyme that is called aldolase splits fructose 1 and 6 bisphosphate into 2 trios that are glyceraldehyde triphosphate and dihydroxyacetone phosphate. So we can say the enzyme that is called aldolase splits F16BP that is called fructose 1,6 bisphosphate into 2 3-carbon molecules. One of them has ketone as functional group and one of them has aldehyde as functional group. Dihydroxyacetone phosphate is an isomer respect to glyceraldehyde 3-phosphate. So DHAP, dihydroxyacetone phosphate, converted into another glyceraldehyde 3-phosphate by the enzyme that is called triose phosphate isomerase. And after that, glyceraldehyde 3-phosphate by using another enzyme that is called glyceraldehyde 3-phosphate dehydrogenated. So this means remove hydrogen from glyceraldehyde 3-phosphate or G3P, and this hydrogen transferred to NAD that act as co-enzyme in this reaction. NAD so gain its hydrogen and reduce to NADH. Simultaneously, an organic and inorganic phosphate also that here is added and creating 1 and 3 bisphosphoglycerate. So this means triose here oxidized to an acid with 3 carbon or contain 3 carbon, that is called 1,3-bisphosphoglycerate that contain 2 phosphate group that added in first and third carbon of phosphoglycerate or glycerate that act as acid. So after that, there is another enzyme that is called phosphoglycerate kinase. That this enzyme by using of this enzyme, 1 phosphate group add to ADP for producing ATP. But because after the fourth step, we have 2 molecules that contain 3 carbon, can be triose or an acid, because this process leads to formation an acid that is called pyruvate. So this molecule contains 3 carbon, and phosphoglycerate kinase add 1 ADP, 1 phosphate group to ADP for producing ATP molecule. And in this step, 3 phosphoglycerate produce. And the enzyme after that, the enzyme that is called phosphoglycerate mutase move the phosphate to a new position, forming 2 phosphoglycerate, and the enzyme that is called enolase removes a water molecule, creating phosphoenol pyruvate PEP. That is a very high energy molecule, After that, the enzyme that is called pyruvate kinase transfer phosphate group from phosphoenol pyruvate to ADP, forming ATP and pyruvate. So, since this happened twice, we generate two more ATP molecules. So, we can say this process is an example of catabolism, that the biological breakdown of complex substance in this reaction is glucose into simpler ones, that is pyruvate. A set of 10 water-soluble systolic enzyme catalyze the reaction, constituting the glycolytic pathway. Glyco, this means sweet, and lysis means split, in which one molecule of glucose is converted into molecule of pyruvate, that is acid. All the reaction intermediate produced by this enzyme are phosphorylated, that means have phosphate group, and we can see in all molecule in this page. So all of them are phosphorylated and water soluble, that is called metabolic intermediate. In addition to chemically converting one glucose molecule into 2 pyruvate, the glycolytic pathway generates 4 ATP molecules by phosphorylation of 4 ATP into ATP. So ATP is formed directly through the enzyme catalyzed joining of ADP with inorganic phosphate that is derived from phosphorylated metabolic intermediate. This process is called substrate level phosphorylation. That is very important, that the ATP in this process that is called glycolysis is produced directly and by substrate level phosphorylation. And in this process, 2 NAD plus is consumed, 1 glucose molecule consumed, 2 NADH produced, and 4 ATP molecule produced, but 2 ATP molecule at the first consume. So the net gain of ATP is 2 in this reaction.